Assalamu alaikum everyone. My name is Mohammed Dawar Zahid, and I think I should, stop introducing myself in every single video. So I'll just straight get into it. So in this video, we'll be talking about the cell surface membrane, the cell wall, and also the centrioles. The three C's. They're pretty simple in A levels. लेकिन अगर आप बात करें अपने AKU और MD CAT और उन सारे exams, your pre-medical exams, so in those exams you do go into a lot of detail, and even in A levels, like, you do go into detail with the cell surface membrane, but wo ek dusra chapter, that's your chapter number 3. So we'll be talking about this over there. So let's just get straight into it. Pretty simple. The first thing that I want you guys to understand is that, and it's pretty simple, cell surface membrane jo hota hai, it's going to give you, essentially, it's going to separate the cell from the surrounding environment. Pretty simple, right? Lekin at the same time, we want it to be partially permeable. So we want that, okay, certain things can move in, certain things can move out. But for example, we don't want the nucleus moving in. If the nucleus moving out, sorry. So if the nucleus moves out, हमारा तो सेल खत्म हो गया, like हमें तो फायदा ही नहीं होना फिर, ठीक है. And similarly, let's say we have lysosomes. So I don't want my lysosomes to be inside the cell. If I have a lot of lysosomes inside the cell, they might, set each other off, and then the entire cell will be destroyed, because just like we talked, the lysosomes, they contain hydrolytic enzymes. They're really, really, scary enzymes. So they can just ruin the entire cell. So we just want, okay, they're made, we just want to push them out. So it's partially permeable. The second thing is, pretty simple, but I'll just still write it, that it controls the movement of things inside and outside the cell. Controls movement. Oh my god, what is wrong with my spellings? But controls movement. Okay, the third thing, and this is a pretty simple thing, but might confuse you a bit. Cell recognition. Okay, so the body has a system of recognizing that, okay, this cell is my cell, this cell is not my cell, and that's pretty important if you're talking about various pathologies. If let's say a person has an infection to the body has to know that, okay, this cell is not my cell and this cell is my cell, and that is my cell recognition systems. So I have these specific proteins, specific glycoproteins on the surface, and these are essentially responsible for allowing me to for giving the cell an identity. And I would like to explain this with the example of a license plate. In the same way how a car has a license plate, so a traffic constable, he just looks at it and he's like, okay, this car is registered, this is a car that is this person can drive this car in this province without any problems. तो वो कहते हैं कि okay, ठीक है. So that's the same thing over here. चौथी चीज मेरे पास है receptor sites. Okay, so the cells, as you guys know, they're not existing in a vacuum, right? They're existing in tissues, they're existing in the entire, in an organ, and then obviously organ systems, and then the entire body. So let's say you're talking about the cell, and the cell is, let's say कि ये वाला जो cell है, it's in the brain. Let's say it's in the medulla of the brain, and now let's say कि the body is secreting hormones. Let's say मेरी, let's say कि specific organ has secreted a hormone, and now this hormone has to go to this cell, the cell in the brain, and it has to signal it. तो इसके लिए मेरे पास ये इस तरह के, ठीक है, I've made it in the same colors because sort of like same चीज़ ये होती है ये, ठीक है. मेरे पास proteins, glycoproteins वगैरह मौजूद होते हैं, and they essentially take this signal and then they trigger an intracellular response, and we'll be talking about that in a lot of detail in your A2, जो हमारे पास CMP और वो cyclic AMP, वो सारी चीज़ें आ जाती हैं, और signaling pathways आ जाते हैं, cascades आ जाते हैं. So अभी के लिए just know what, just know that we have these receptor sites present, ठीक है. इसके साथ साथ पांचवी बात मेरे पास ये है कि आई हैव माई माइक्रोविलाई प्रेजेंट. अब मेरे माइक्रोविलाई क्या होते हैं? माइक्रोविलाई ना, आई हैव मेड लाइक अ प्रीटी हॉरिबल ड्राइंग ऑफ इट, लेकिन माइक्रोविलाई आर दीज एक्सटेंशंस ऑफ द सेल सरफेस मेम्ब्रेन, एंड व्हाट दे डू इज दैट दे इंक्रीज द सेल सरफेस एरिया, ठीक है. सो मुझे लार्ज सरफेस एरिया इन सर्टन केसेस मुझे चाहिए होता है. फॉर एग्जाम्पल, अगर लेट्स से सेल इंटेस्टाइन के अंदर है, या लेट्स से सेल किसी भी जगह पर जहाँ पे उसको लार्ज सरफेस एरिया की ज़रूरत है, तो उसके लिए इस तरह के मेरे साइटोप्लाज्मिक extensions बन जाएंगे. And again, please understand, this is really rudimentary. In reality, it's really different, ठीक है. लेकिन जो आपको वो diagrams वगैरह देंगे आपके paper 1 और आपके paper 2 में, they're going to be pretty rudimentary, ठीक है. बहुत आसान होंगे. I know, like now that I'm looking at it, it sort of looks like a duck, like duck का face लग रहा है. खैर, okay, so moving on, the sixth thing, and this is important, that is that we have components. These are actually our glycoproteins and glycolipids, ठीक है. Conjugated molecules होते हैं, components that that cause cell adherence. Let me explain. This is a pretty simple topic नहीं, concept. अगर let's say मेरे पास ये cell है और मेरे पास ये cell है, अब they're not existing in a vacuum, right? they are existing in a tissue. So I want that okay, both of these they need to be connected. तो मेरे पास कुछ proteins से होते हैं, कुछ conjugated proteins होते हैं, if you want me to be more specific, that are sort of joining them together. ठीक है, और ये सिर्फ इन दो सेल्स को नहीं ज्वाइन कर रहा है, पूरे मेरे टिश्यू को ज्वाइन कर रहा है, पूरे मेरे ऑर्गन को ज्वाइन कर रहा है. ठीक है, and we'll be talking about this in our cell membrane वाला एक पूरा chapter in which there's only us talking about the cell membrane, all of those things. So अभी के लिए हम इतनी ही डिटेल में जाएंगे and then let's move on to the cell wall. Okay, so coming to the cell wall is a pretty simple structure. I've made a pretty rudimentary cell wall. The first thing is that I have my cellulose fibers. So it's made of my cellulose fibers, but cellulose के साथ साथ I also have these things called hemicellulose and I also have my pectin. ठीक है, तो मेरे पास essentially इन तीनों चीज़ों का बना हुआ होता है. ठीक है, if I talk about my cellulose fibers, there's a really common question that comes in your paper too, which is that describe the cell walls relation, the cell wall structure in relation to the cellulose that is present over there. ठीक है, so इसका आंसर ये है कि हमारे पास cellulose microfibrils होते हैं. I'll explain this in a second. ठीक है, which contain polysaccharide cellulose. थोड़े से हम लोग अभी लिमिटेड हैं, we can't go into a lot of detail, but just like I mentioned, important topic for your आगे के एग्जाम्स, and I'll be making a video on it as well. लेकिन अगर मैं इसको zoom in करके आप लोगों को दिखाने की कोशिश करूँ, if I make a sort of like a cut of the cell wall, तो it's essentially going to be these microfibrils, sort of if you have ever seen a construction site, तो वो steel beams लगाते हैं, steel beam के अंदर वो concrete pour करते हैं. So the cellulose microfibrils, these are those steel beams. ठीक है, और polysaccharide cellulose is that concrete. ठीक है, so pretty simple. इसके साथ साथ अगर हम अब आगे बात करें तो हमारे पास एक और स्ट्रक्चर प्रेजेंट होता है जिसको मैं नाम देता हूँ अपना मिडल लेमेला का. मिडल लेमेला डज नॉट मीन दैट कि जी सेल वॉल के दरमियान में कोई चीज़ है. ऑल इट मीन्स इज, एंड अगेन रियली सिंपल, उसमें हम अभी बात कर रहे हैं, हम डिटेल में नहीं जा रहे. ये मेरे पास दो सेल्स हैं. ठीक है, यू कैन सी देयर सेल वॉल्स. तो इसके दरमियान में एक पतली सी लाइन होती है मेरे पास, उसको मैं नाम देता हूँ अपने मिडल लेमेला का. ठीक है, so this is going to be my middle lamella, and what it does is that it sort of cements adjacent cells together. ठीक है, just a second, I'll have to pause. I'll just write cements adjacent cells. Okay, so it's going to basically cement the adjacent cells together. Not a lot of detail right now, we'll talk about it in the future. Okay, so functions of this cell wall, and pretty simple. The first one is that we are going to have it's going to protect the cell from bursting. Basically अगर मेरे पास सेल में पानी बहुत ज़्यादा बढ़ जाता है तो वो पानी एक प्रेशर जनरेट करेगा. ठीक है, दैट ऑस्मोटिक, इट्स ऑल्सो कॉल्ड ऑस्मोटिक प्रेशर, हाइड्रोस्टैटिक प्रेशर, टर्गर प्रेशर. ठीक है, टोनोप्लास्टिक प्रेशर, लॉट ऑफ नेम्स. ठीक है, सो दिस सेल वॉल इट्स नॉट गोइंग टू अलाउ द सेल टू बर्स्ट. सो दैट्स प्रीटी इंपॉर्टेंट. द सेकेंड थिंग दैट वील हैव इज दैट इट्स गोइंग टू गिव स्ट्रेंथ टू द प्लांट एज अ होल. So if you, let's say, take my hand for example, and my hand is going to be pretty soft compared to, let's say, a plant. So एक plant की stem पकड़ लो, मेरा हाथ पकड़ लो, even though like I do go to the gym, I do try to have like good biceps and all, but still a plant is going to have better ones. ठीक है, it's not that I'm not doing good at the gym, I hope I am. खैर, so एक और बहुत important बात कि it allows water to pass through. And about this again, we'll be talking about this a lot future वाले chapters के अंदर. लेकिन अभी के लिए इस तरह समझो, इसमें छोटे छोटे से ना होल से होते हैं और पाथ से बने हुए होते हैं. तो हमारे पास जो पानी होता है वो उनमें से पास थ्रू करके, लेट्स से मैं इसका एग्जांपल दूँ, टिश्यू है तो यहाँ पे छोटे छोटे होल से बने हुए, तो दैट वाटर इज एबल टू ईजिली पास थ्रू. सो दैट इज़ व्हाट हैपन्स. Now the last thing that we're going to talk about is going to be these things called, these structures called plasmodesmata. Let's use a different color. It's pretty important, plasmodesmata. Okay, so plasmodesmata, I know it's like a fancy name, but it's pretty similar. Okay, it's pretty simple. So essentially what I want you guys to sort of understand, let me just make, you know, two lousy looking cells. Okay, so it's sort of like this. It's this cytoplasmic, so it's exactly a just a second, it's a small, it's basically small thin strands of cytoplasm. Essentially these are अगर इस सेल की मैं बात करूँ, इस सेल में एक सेकेंड, सॉरी, ओके, सो इस सेल में मेरा जो okay, एक सेकेंड, this is too big. Okay, so cytoplasm over here, this cytoplasm over here, so यहाँ पे भी it's going to be continuous with the cytoplasm. So it's sort of like, you could say it's making it, you know, one whole cell. You could say that, like, not really true, but you know, I could sort of say that. Okay, the important thing about it is that it allows very, very quick movement of water and mineral ions, etc. And all of those, you know, things. So हमारे लिए, it's basically, I want you to sort of think of it as a shortcut, and that wraps up the cell wall, and let's move on to the centrioles. Okay guys, so just centrioles, they're pretty similar. They have a bad reputation for being really confusing, but let's just do them quickly. So they're present in almost all animal cells, plus some fungi, some algae, and they're not in plant cells of higher plants. You don't need to know what higher plants exactly are. They'll just ask you that, okay, are they present in plant cells? In normal plant cells of higher plants, you'll say they're not present. Sometimes present. Okay, so not really sometimes, but I would say they're present in certain, not in all. You can't really generalize that, okay, all fungi have centrioles. You know, certain have them. Okay, so the next thing that I want you guys to know is that their dimensions. So they love to ask you their dimensions. So 0.5 micrometers in length and 0.2 micrometers in diameter. Okay, अब बात आती है, अच्छा centrioles हमने कर लिए, microtubules क्या होते हैं? Centrioles are made up of these microtubules, and these microtubules in return are made up of a globular protein called, जिसका नाम होता है आपका, it's pretty simple, it's tubulin. Microtubules and tubulin protein. Okay, अब ये जो पूरा स्ट्रक्चर है, इसको मैं नाम दूंगा सेंट्रियोल का, इसको मैं नाम दूंगा सेंट्रियोल का. इस सेंट्रियोल में, do you guys see these small, you know, linear-ish proteins, and they're sort of forming these microtubules. They're forming these triplets, 1 2 3, ठीक है, and let's just count them, 1 2 3 4 5 6 7 8 9. So we will say, and this is a line that you have probably seen a lot in your textbooks, that centrioles are made up of, let's just write it over here, they're made up of 9 triplets of microtubules. A lot of questions come इस चीज़ के बारे में, ठीक है. So now we are done with this. अब इसके साथ साथ एक मैंने important बात समझानी है. For diagrams, they're lying right in a right angle configuration. This angle is right angle. So they could also be lying sort of, you know, like this, ठीक है. As long as the angle between them is 90 degrees, ठीक है, it's going to be a centriole, ठीक है. Because understand that हमारे सेल में और भी बहुत किस्म के, you know, प्रोटीन के like filamentous structures पड़े हुए होते हैं, प्रोटीन के इस तरह के सिलेंडर्स पड़े हुए हैं, बहुत ज़्यादा चीज़ें होती हैं, ठीक है. तो ये बात प्लीज़ याद रख लो कि अगर वो 90 डिग्रीज पे पड़े हुए आपस में, तो they're going to be centrioles. Okay. And now moving on to their functions. Now their functions, they bother kids a lot, because the problem is कि ये वाले functions ना हम लोगों को ने अभी पढ़े नहीं होते. So for a kid who is freshly, you know, who's like a fresh A1 kid, अभी start किया है, कुछ महीने पहले वो levels ख़त्म किया था, उस बच्चे ने चीज़ें नहीं पढ़ी हुई होंगी, ठीक है. तो functions of microtubules. So I just want you to, you know, for now unfortunately you'll have to memorize them for your exams. तो जो midterms वगैरह भी आ रहे हैं दिसंबर में, तो उसमें ये टेस्ट होगा, तो प्लीज़ अभी याद कर लेना. लेकिन moving on, I'll be, you know, because एक एक लाइन का पूरा चैप्टर है, ठीक है. So we'll be talking about that. The first thing is, जो centrioles होते हैं, ये हमारे spindle fibers बनाते हैं, and this process is involved in our न्यूक्लियर डिविजन. ओके, सो जब हम क्रोमोसोमल डिवीजन कर रहे होते हैं, स्पेसिफिकली क्रोमोसोम्स को डिवाइड कर रहे होते हैं, तो हमारे पास स्पिंडल फाइबर्स बन रहे होते हैं. सेकंड बात ये, हमारे पास बेसल बॉडीज़ होती हैं. ठीक है, सो माइक्रोट्यूब्यूल्स and centrioles, let me just write it over here, /centrioles, they're responsible for all of these things. ठीक है, so basal bodies होती हैं. basal bodies का रोल क्या होता है कि basal bodies are involved in the formation of cilia and flagella. तो ये भी प्रोटीन्स वाले स्ट्रक्चर्स होते हैं, तो ये भी वही हमारे पास बना रहे होते हैं. इसके बाद अब मेरे पास एक साइटोस्केलेटन होता है, एंड साइटोस्केलेटन के लिए इन द नेक्स्ट वीडियो आई विल बी एक्सप्लेनिंग यू गाइस साइटोस्केलेटन क्या होता है. ठीक है, इट्स बेसिकली एन इंटरनल स्केलेटन ऑफ द सेल. ठीक है, इट्स रियली इंटरेस्टिंग एंड विल बी टॉकिंग अबाउट दैट. अभी जो साइटोस्केलेटन है ना, यही बेसिकली दो काम कर रहा होता है. एक तो ये सॉर्ट ऑफ एक्ट्स लाइक अ हाईवे, सेल में हाईवे सी बन जाती हैं. सेकंड व्हाट इट डज इज दैट इट्स गिव्स मी सपोर्ट. मेरे सेल को पूरा, क्योंकि साइटोस्केलेटन है, इट्स गिविंग मी सपोर्ट. ठीक है, एक ये चीज़ आ गई. एंड लास्टली व्हाट आई एम गोइंग टू टॉक अबाउट इज दैट मेरे जो माइक्रोट्यूब्यूल्स होते हैं, लेट मी यूज पिंक. ओके, सो माई माइक्रोट्यूब्यूल्स आर ऑल्सो गोइंग टू बी इन्वॉल्व इन द मूवमेंट ऑफ द सेल एज अ होल. And the reason why I'm emphasizing on this so much is because they love to say, okay, you know, I gave a poison to, and I'm talking about your AKU test MCQs as well. So they'll say that I gave a specific poison which damaged the microtubules. Which of the following processes will not be affected? They'll give you these. इनमें से 3 दे देंगे, ठीक है, और एक कोई random वाला डाल देंगे. So it's really, really important. I'll even mention it over here that this is going to be pretty important for you guys. And this wraps up our videos on the 3 C's, the cell wall, the cell surface membrane, and the centrioles. The next video is going to be the last video of this chapter. I'm honestly really excited. I do not like this chapter. और वो हमारी अगली वीडियो, लेकिन इट्स गोइंग टू बी अ फन वीडियो, सो प्लीज स्टे ट्यून फॉर दैट. एंड इसके बाद लेट मी नो इन द कमेंट्स व्हिच चैप्टर यू वांट मी टू डू नेक्स्ट. आई वाज थिंकिंग ऑफ डूइंग like biological molecules because I do sort of want to get that out of the way, but I would love to hear your opinion on it, so please do let me know. And please, as always, share, subscribe, like, all of those things. And that's it for today. Thank you so much.